Today we are going to start a new topic, the structure of the prokaryotic cell. You can also say this is the structure of the bacteria. So in exam, if the examiner asks you about structure of the bacteria, you can tell them easily after studying from me. Alright. So we are going to draw the structure of this. Alright, let's do this. So look carefully how I will be doing it. First we will draw the structure of this bacteria. Right? Okay, now we are going to do this. I think this is good, right? Alright, these were the ribosomes. Now we are going to draw the circular DNA. This is the circular DNA. Now we are going to draw the naked DNA. So that's how it looks like. Alright, you can say this is the naked DNA. Now there is another structure, it is not in our syllabus, but we can also learn that. It's called sex pili. Alright, it's called the sex pili. It also have these lines in it. Right? Say this is the sex pili. Now let's learn about every structure that I've drawn here. Is the cytoplasm. Where is the cytoplasm? Alright, this is the cytoplasm. Cytoplasm. Alright? Now this is the circular DNA. So let's label the circular DNA. Circular DNA. And it is also called plasmid. Alright? So in exam, when we are going to study our last chapter, the miscellaneous biology, so bacteria will play an important role over there. Now this is the naked DNA. Right? The bacteria has two types of DNA in it. So when we are going to learn about the human insulin, it is going to be really important over there. This is the naked DNA. It's also called the core DNA. Alright, these were here. Alright. Now I told you that this is the mimosome, but I missed one structure over here. This is the structure. Right, this is the mimosome. I mistakenly told you that this is the mimosome. This is not the mimosome, it's called flagellum. Alright, that was a mistake done by me, so forgive me. as this is the mimosome, and this is not even in your syllabus. This gentleman right here is the flagellum. So the purpose of the flagellum is locomotion. It helps the bacteria to move. Flagellum, locomotion. Its main purpose is to help the bacteria to move. Now we talked about sex pili. It's not in your syllabus, but you can know that sex pili helps to exchange genetic information. Alright, I'm not going to write its function, but you can just remember it that it helps the bacteria for exchanging genetic information. Alright, then we have ribosomes over here. Ribosomes. Ribosomes. Then we have the cell wall. This is the cell wall. And lastly, this is the cell membrane. In our last class, we discussed what is a cell membrane and a cell wall. Alright, as we have talked about the cell membrane in our last class, here we have talked about the cell membrane. Great discussion we did about cell membrane. So this comes to an end of our structure of the bacteria, and now it's complete. We have completed everything, but there's one thing left: the size of the ribosomes. You guys must know what is the size of the ribosomes. So the size of a ribosome is 70S. Remember that. Alright, so our next topic is specialized cells. So there are specialized cells in humans and in plants. Alright, specialized cells. Let's do specialized cells. Specialized cells. Alright, so let's begin. I'm going to give you an introduction to what does this mean. So we are going to be doing in human, in plants, alright, in humans and in plants. So let's begin. Okay. So in humans, first we have ciliated cell. We have ciliated cell. Then we have RBCs. Then we have ovum or egg. Alright, in plants we have root hair cell. We have palisade cell. Then we have xylem. Then we have phloem. Both of these structures are really similar. I will be doing them side by side. Alright. Then we have the last topic, which is levels of biological organization. Levels of biological organization. Biological organization. Alright, so this was the introduction. So we will be doing our first topic now, which is specialized cells in humans. So the first specialized cells in human is the ciliated cell. Alright, ciliated cell. Let's begin. Alright, specialized cells in human, they are ciliated cells. Alright, so first I am going to draw the structure of the ciliated cell, and then I am going to make you guys understand what it is. Alright, so let's begin. Alright. Alright, I think I made a mistake over here. Let me check. You guys may not be able to understand a difference between one thing, so I will be drawing it red. Alright, so let's label it. This right here is the goblet cell. This right here is the goblet cell. What you see there is a nucleus. Alright, and now this entire structure structure is the ciliated cell. Alright, now these are the brush borders. These are the brush border. Alright, now what does it do? So the brush border is the cilia. They move mucus out of body. They move mucus out of the body when we eat something. So when we eat something and mucus, everybody has mucus in their throat. So cilia moves mucus out of the body. All right. This is the main purpose of the cilia. These brush borders. That's what they do. This is the goblet cell. Goblet cell produces the mucus which is in our throat. Which is goblet cell produces that mucus. All right. Now, how this helps us? We are going to draw a simple structure which may represent us our throat. Right? Alright. Alright, so now we are going to write the definitions. First is a goblet cell. Goblet cells. Goblet cells produce sticky chemical called mucus, which traps dust particles from entering in the airway. Now ciliated cell, I just told you the brush borders are the cilia piping, alright? These are the ciliated cells. Now the ciliated cells via rhythmic wiping movement of cilia wiping pushes the mucus in upward direction, from where it is either swallowed or spit out. Alright, so this comes to an end of our first human structure. Some of you might not have understood till now that what does this do. So I may draw a simple diagram to make you understand. Right? So you can imagine that this is your throat. Alright, so now we are going to draw a second layer. Alright, now you can imagine that this is the goblet cells. Alright, now this is now these are, now you will see the black color, so I will draw it with yellow. Alright, you can imagine these as the ciliate cell. Alright, these are the ciliate cell, the brush borders you can see. Alright, now if a person smokes over here, there is a person who smokes. Alright, as they smoke, these brown dust particles enter their throat, enter their. Alright, so what is going to happen, these cilia are going to produce, the goblet cells are going to produce the mucus, and the mucus is going to trap all of these dust particles or the smoke particles, whatever you say they are, or the carbon dust. Then the cilia, these yellow brush borders, are going to either let you swallow this or spit out. Alright, sometimes you spit out the mucus, right? Or sometimes you swallow it. That's how it is. Alright, that's it. Here the first structure comes to an end. The second structure we have is the red blood cells. Alright, so we will be doing the red blood cells now. Red blood cells. We can also call them RBCs. Alright, so the RBCs we are going to do tomorrow. Alright, that's it for today. Allah Hafiz. Ciao.